Transcription. In transcription, an mRNA copy of a gene is made from DNA. In eukaryotic cells, transcription occurs in the nucleus, but prokaryotes don't have a nucleus, so transcription takes place in the cytoplasm. There are 4 stages to transcription. So the first one is that RNA polymerase attaches to the DNA. So transcription starts when RNA polymerase attaches to the DNA double helix at the beginning of a gene. The hydrogen bonds between the 2 DNA strands in the gene break, separating the strands, and the DNA uncoils at that point, exposing the bases. One of the strands is then used as a template to make an mRNA copy. 2. Complementary mRNA is formed. The RNA polymerase lines up free RNA nucleotides along the template strand. The free bases are attracted to the exposed bases. Specific complementary base pairing means the mRNA strand is a complementary copy of the DNA template strand, except the base T is replaced by U in RNA. Once the RNA nucleotides have paired up with their specific bases on the DNA strand, they're joined together by RNA polymerase, forming an mRNA strand. 3. RNA polymerase moves down the DNA strand. The RNA polymerase moves along the DNA, assembling the mRNA strand. The hydrogen bonds between the uncoiled strand of DNA reform once the RNA polymerase has passed by, and the strands coil back into a double helix. So you can see here it's unwound. As it actually transcribes that part, it will rewind up to form the double helix again behind it. 4. RNA polymerase reaches a stop signal. When RNA polymerase reaches a sequence of DNA called a stop signal, it stops making mRNA and detaches from the DNA. In eukaryotes, mRNA moves out of the nucleus through a nuclear pore and attaches to a ribosome where translation, which is the next stage of protein synthesis, takes place. Editing mRNA. So transcription produces different products in eukaryotes and prokaryotes. In eukaryotes, the introns and exons are both copied into mRNA during transcription. mRNA strands containing introns and exons is called pre-mRNA. In a process called splicing, the introns are removed and the exons, which are the coding parts of the mRNA, are joined together, forming an mRNA strand. So the exons are the bit we actually want. So the introns are removed by splicing, and then you get the mRNA strand with just the exons in. This happens in the nucleus, and the mRNA then leaves the nucleus for translation. In prokaryotic mRNA, the mRNA is produced directly from the DNA without splicing because there are no introns, or there's no non-coding DNA. So there's no introns in prokaryotic DNA. So that is it for transcription.